talking a little bit about the microbial community and how the community is responding to the infusion of oil and gas. I mentioned last week that we had the initial hydrocarbon results back. I want to make a couple of comments on those numbers as well. The levels of hydrocarbons within about 3 kilometers of the wellhead.
I can sign for that, Donald. Donald?
I just asked.
Hi. How are you?
Can you hit your mute button, please?
I'm just listening.
You're listening, but your mute button isn't muted. Okay, thank you. The hydrocarbon levels were on the order of parts per million, close into the wellhead within 3 kilometers. Once we got about 5 kilometers out, the levels decreased to parts per billion range, anywhere from single digit to 10 to 100 ppb. So we saw a decrease, as you would expect, in hydrocarbon concentrations with increasing distance from the leaking wellhead. I've requested additional analysis be done on those samples, and we'll quantify the concentration of different components of hydrocarbons, primarily the PAHs and the biomarker, the labile aliphatic components. So with respect to the microbial community and how it's responding to the oil spill and the gas input at depth, we've been doing several different types of experiments and measurements to evaluate the microbial community response. And what I want to briefly talk about today is just microbial cell abundance and how it varies with depth through the water column and how it changed during short-term incubation experiments that we did. The microbial abundance, so the numbers of microorganisms, are larger within the deep water plumes than they are above and below the plume features. We haven't completed these analyses yet. They're still ongoing, but the data that we have so far shows about a 2-fold or more increase in microbial numbers in the layers associated with the plume versus the layers above it and below it. More strikingly and actually quite almost shockingly, we did 24-hour incubation experiments to measure rates of oxygen respiration, and we measured some of the highest rates of respiration that I've ever seen anywhere. And so I guess I wasn't so surprised by the fact that in some cases, especially close in to the wellhead where the concentrations of methane and other hydrocarbons were highest, the microbial numbers almost doubled in a 24-hour period. That's really, really surprising. And these numbers were determined using a flow cytometer, which is a pretty sensitive way to determine microbial abundance. And we're also doing direct cell counts with stains to sort of have a separate way of determining the change in cell numbers over time. But clearly the microbial community is responding rapidly and rigorously to the input of oil and gas. And as we get more data, I think this picture will become more complete. But I just wanted to share the fact that we are seeing fairly dramatic changes in cell numbers. We don't know what fraction of the population is responding to the oil and gas, just that the total population is changing. Melissa Crespo-Medina, who's a postdoc in the lab, has been doing the direct microscopic counts, and just in terms of the descriptions of the cells, you see very big, fat, happy cells that look a lot like methane oxidizing or methane eating bacteria, but we'll be doing some specific staining to identify those organisms. But they're pretty characteristic in terms of what they look like. So I'm pretty convinced that the methanotrophs are some of the ones that are responding to this input of oil and gas. So with that, I will stop, and I'm happy to take any questions that anybody has.
Could you characterize, the fact that methane-eating bacteria are apparently, obviously developing at the site or at the site, is that something that is good news or bad news on the scale?
Well, it's good news in that these organisms they're going to consume the methane, which is good. The fact that they're growing, I certainly don't expect growth of that magnitude to be sustained over even more than a few days, because they're going to run out of nutrients. But certainly this tells us that the organisms that are there's a fraction of the population that's responding very rigorously and rapidly to gas and oil inputs. These organisms are growing very rapidly. I should say that there was no significant change in cell abundance in the respiration measurement. The incubations that we did at the control site, where there was no oil and no gas, the cell numbers didn't change over a 24-hour period, and the respiration rates were about a factor of 25 lower. So, this is a response that is distinct to the areas where the oil and gas are being input into the system. The fact that these organisms are responding is good, definitely. Like I said, we don't know what fraction of the population is responding. Certainly the methanotrophs are, but probably other organisms that consume alkanes like ethane and propane or butane. Oil stimulates microbial growth as well. My colleague Andreas Teska at the University of North Carolina at Chapel Hill had a postdoc doing some experiments with just surface water, where they added oil to the surface water, and within 2 days they could see a dramatic change in cell abundance and enzyme activity. So the impact on the microbial community is effectively instantaneous. The question is, how long can it be sustained, and what are the repercussions for things like oxygen concentration and nutrient concentrations? And again, we don't know the answers to those things yet. We have experiments that are spinning up in the lab that will help us sort of, I think, fine-tune our understanding of these processes and how what their temporal evolution is.
Any sense of what this is or might do further up the food chain?
Not at this point. I mean, I think, we didn't make any measurements of higher trophic levels, and so I don't really feel comfortable speculating on how that is going to cascade. In the August cruise, we'll be doing measurements of zooplankton and fish and other, higher levels of the food chain, so we'll be better able to answer that question after that cruise is done with.
Can I ask a question from the phone?
Sure.
Hi, this is Jeff Ball at the Wall Street Journal. I was just curious to see if you could compare the state of your results so far with the results that are coming from the government. There was a report from the JAG, I think, which is the Joint Assessment Group, I think is the acronym, a week or so ago, which looked at results on oxygen levels through, I think, late May. Mm-hmm. And essentially, I'm sure you're familiar with that report, but essentially it found a reduction in oxygen levels, but not a reduction to levels that would alarm the people who analyzed that. And I'm just trying to understand what you found in the context of that and other studies that you think are relevant.
Okay, the question is how did the oxygen levels that were reported in the JAG report from a couple of weeks ago compare to our results? To some extent, that's comparing apples and oranges. The JAG report focused primarily on stations that were very, very close to the wellhead. A lot of that data, I believe, was from the Brooks McCall, which is tasked with tracking the dispersant plume, which is, that's within 3 kilometers of the wellhead. And we did not see consistent oxygen drawdown that close in, and I wouldn't expect to, because the water is very young. The plume is very young when it's that close to the wellhead, and we didn't see significant drawdowns in oxygen. And by significant, the bottom water has around, say, 7 milligrams of oxygen per liter, and we were seeing levels down to 4 milligrams per liter and sometimes below that, but not until we got 8 to 10 kilometers away from the wellhead. So the water has to age, and the microbial processes have to sort of kick in and start consuming and making a dent in that oxygen pool.